Dear students, welcome to all of you. Today we are here to discuss morphology, anatomy and reproduction of genus Pteris of pteridophytes. The name Pteris is derived from pteron, a feather, due to the feathery appearance of the fronds. The English name fern is a contraction of the Anglo-Saxon piperon, that is a feather. In some parts of England, it is called King Charles in the oak tree. In Scotland, it is said to be an impression of the devil's foot, and yet witches were reputed to detest this fern for the reason that it bears on its cut stem the Greek letter X, which is the initial of the Christos. In Ireland, it is called the fern of God, because if the stem is cut into 3 sections, on the first of these will be seen the letter G, on the second O, and on the third D. So far the systematic position of genus Pteris is concerned, it belongs to kingdom Plantae, phylum Pteridophyta, class Phelicopsida, order Polypodiales, family Pteridaceae. The genus Pteris is a large and diverse genus of about 280 terrestrial and epiphytic species of ferns of predominantly tropical and subtropical regions. The species grow in moist, shady forests and on hillsides. Many species are found on the roadsides and valleys of western and northwestern Himalaya. The common species in India are Pteris vittata, Pteris critica, Pteris biaurita, Pteris quadriaurita, Pteris stenophylla, and Pteris welchiana. The Pteris vittata is a low-level fern and brings out new leaves throughout the year. It is very common along mountain walls and grows up to 1,200 meter altitude. Now the morphology of the sporophyte. The dominant stage in the life cycle of Pteris, constituting the main plant body, is the sporophyte. It is differentiated into roots, stem and leaves. The primary root formed from embryo is short-lived and is soon replaced by the adventitious roots. These roots are cylinder, black, wiry, and arise from ventral side of rhizome or from all over the surface. The stem is perennial, rhizomatous, underground, branched, and covered by brown scales. Some species have persistent leaf bases on the rhizome. The leaves are macrophyllous, unipinnate, and arise acropitally on the rhizome. Bipinnate leaves are seen in Pteris biaurita. Decompound and digitate leaves are also present. Fully developed leaves are known as fronds. The petiole base is covered with brown scales and sometimes with ramenta. The rachis has several sessile, lanceolate, coriaceous leaflets arranged in pairs except the terminal one. The leaflet is broader at the base and gradually tapers towards the apex. The middle leaflets are larger and the other leaflets show gradual decrease in size towards the basal and apical regions of the rachis. The leaflet is top, has a midrib from which arise the lateral veins with dichotomous branching. The venation is open dichotomous. The younger leaves show circinate vernation. Growth rate is very slow. Now the anatomy of sporophyte. First, the anatomy of rhizome. In transverse section, the rhizome is oval in outline and differentiated into epidermis, cortex, and stele. The epidermis is single-layered with quadrangular cells. The epidermis is covered by thick cuticle. The cortex is multi-layered and differentiated into 4 to 5 layered sclerenchymatous hypodermis and inner multi-layered broad parenchymatous region. The parenchymatous region has root traces and leaf traces. The stellar structure differs from species to species. In Pteris vittata, it is dictostele with a ring of vascular strands that is meristeles. Meristeles are embedded in the parenchymatous ground tissue. Each meristele is elliptical with single-layered endodermis having Casparian strips in its radial walls. 1 to 2 layered thin-walled pericycle is present below the epidermis and surrounding the phloem. Phloem has only sieve cells and phloem parenchyma. It completely surrounds the xylem. Xylem is present at the center of meristele. It consists of tracheids and xylem parenchyma. Now the anatomy of rachis. In transverse section, the outline of rachis or petiole is semicircular. It is differentiated into epidermis, ground tissue, and vascular bundle. The epidermis is single-layered with narrow quadrangular cells covered by thick cuticle. Some epidermal cells give rise to uniseriate bicellular hairs called as ramenta. The ground tissue has multi-layered sclerenchymatous hypodermis, followed by parenchymatous tissue in which the vascular bundle is embedded. The vascular bundle is V-shaped or U-shaped and embedded in the parenchymatous ground tissue. Pericycle is 1 or 2 layered and parenchymatous. Xylem is at the center with meso-arc condition surrounded by phloem. Now the anatomy of leaflet. Leaflet has epidermis, mesophyll, and vascular bundle. The epidermis is single-layered, present on both the upper and lower surfaces. It has stomata only on the lower epidermis. The mesophyll is either homogeneous or differentiated into upper palisade and lower spongy with broader intercellular spaces. Hypodermal region of midrib has sclerenchymatous strips on both abaxial and adaxial sides. Midrib has single concentric amphicribal vascular bundle surrounded by single-layered pericycle and endodermis. The vascular strands are embedded in the mesophyll. Now the root anatomy. Root is circular in outline and distinguished into epidermis, cortex, and stele. The epidermis is single-layered with thin-walled cells. A few cells form the root hairs. The cortex is multi-layered and differentiated into parenchymatous outer cortex, sclerenchymatous inner cortex, and a single-layered endodermis with Casparian thickenings. The stele has single-layered thin-walled pericycle, central plate-like exarch and diarch xylem surrounded on either side by phloem. Now the reproduction of genus Pteris. The Pteris sporophyte reproduces vegetatively and asexually. First, the vegetative reproduction. It takes place by progressive death and decay of older portions of the rhizome. When the decay reaches the point of branching, the main axis and branch get separated and grow as individual plants. Asexual reproduction. The asexual reproduction takes place by spore formation. Genus Pteris is homosporous and produces only one type of spores. Now the sorus structure. The spores are formed in sporangia arranged in sori on a sporophyll. The fertile frond with sporangia is called as sporophyll. Sporangia are formed in groups known as sori. A sorus is developed from a pad of tissue known as receptacle or placenta on the lower surface nearer to the margin of leaflet of sporophyll. Every leaflet has two sori, one on each side. Since the sorus is extended as a narrow line along the margin, except at some length at the apical and basal regions, it is called as linear sorus. It is called as mixed sorus because both young and old sporangia are intermingled. As the sorus has several sporangia, it is actually a cynosorus. Besides sporangia, there are multicellular uniseriate strial structures known as paraphyses, developing from placenta. The sorus is protected by reflexed margin of the leaflet known as the phallus inducium. Structure of sporangium. The sporangium shows leptosporangiate development because it is developed from a single initial. It has a multicellular long cylinder stalk called pedicel or seta and a globose or oval body known as capsule or spore sac. The spore sac has a single layered jacket. Some cells of the jacket are modified to form an annulus and a stomium. Annulus is a ring-like structure with vertically elongated cells. It begins from the stalk on one side, encloses the top, and extends up to halfway on the other side, where it joins with the epistomium of stomium. The annulus cells have thin outer tangential and lateral walls and thickened inner tangential and radial walls. It helps in the dehiscence and dispersal of spores. The stomium is present in between the end of annulus and stalk that forms the remaining portion of the ring. The stomium cells are long, flat, and thin-walled and permit easy cleavage of the sporangium and dehiscence. The middle two cells of the stomium are narrow, radially elongated, and called as lip cells. 2 or 3 cells present above the lip cells form the epistomium, and the 2 or 3 cells below the lip cells form the hypostomium. The other cells of jacket are thin-walled and polygonal or hexagonal. Anterior to the jacket, a 1 or 2 layered nutritive tissue, the tapetum, is present. It encircles the sporogenous tissue. The sporogenous tissue usually forms 12 spore mother cells that divide meiotically and form 48 haploid, morphologically similar spores, that is homosporous. The young sporangium is green while the mature sporangium is dark brown. Now the sporangial dehiscence. At maturity, the capsule is dry. In dry conditions, the annulus contracts and the sporangial wall is broken transversely at stomium between the lip cells. The cytomium bends backwards, carrying the upper half of the broken capsule containing the spores that are exposed for wind dispersal. When the annulus absorbs moisture, it flips forward, hurling the spores with some force and snaps back to its original position. This mechanism of spore dispersal is known as sitting or catapult mechanism. The spores are carried away by wind and remain viable for a longer period. The spores are reddish brown and triangular and have clear triradiate mark known as trilete on their surface. Spore wall has outer thick sculptured axine that is exospore and inner thin smooth intine or endospore. It is uninucleate and unicellular. Now the sexual reproduction of genus Pteris. Each spore germinates and forms the gametophytic phase called the prothallus, which reproduces sexually. The prothallus of Pteris is caudate, dorsiventrally flattened, anterior-posterior with apical notch at the anterior side. It is parenchymatous and multicellular with thickened center that gradually thins down towards the margin. Below the notch, it has a few meristematic cells. The cells are polygonal with protoplast showing primordial utrical condition with disc-shaped chloroplasts and haploid nucleus in every cell. It has unicellular rhizoids at the posterior side on the ventral surface, among which the antheridia are developed. Below the notch, the female sex organs, that is archegonia, are present. The prothallus is usually monoecious but sometimes also dioecious. The sex organs are developed on the ventral surface and exposed directly to moisture, which is needed for the development and dehiscence. The male sex organs are developed earlier than the female sex organs. Hence, prothallus is protandrous. The sex organs are without any stalk and the male gametes are multiciliated. Now the sex organs. First, the antheridia. These are the male sex organs which develop and mature earlier than archegonia. These are small, sessile and globular structures. The antheridial wall consists of 3 tubular-celled jacket. These cells are the first ring cell that covers the basal portion of the antheridial body, second ring cell that covers the middle portion of the antheridial body, and the cap cell that covers the apical portion of the antheridium. The cap cell forms a lid. Apart from these cells, there is a fourth cell at the base that can be considered as a single-celled stalk. The wall encloses 32 spermatocytes. Each one is metamorphosed into a long, spirally coiled, multiciliate, motile antherozoid. Dehiscence of the mature antheridium takes place in the presence of water. The jacket cells and androcytes absorb water, swivel, and exert pressure, due to which the cap cell is either tilted upwards or thrown away. The antherozoids covered by androcyte membrane extrude out in masse. This extrusion is further assisted by the inward swelling and shortening of the wall cells. The membrane around the spermatozoids is dissolved and the male gametes become free to swim. Now the archegonia. The archegonia are developed later than antheridia near to apical notch of the prothallus. It is flask-shaped with a swollen embedded venter in the prothallus and a projected short cylinder, slightly curved neck. It has no ventral wall or jacket. It consists of a basal naked egg cell, above which is a small ventral canal cell. Neck consists of a single layered jacket with 4 vertical rows of cells, with each row having 4 cells. So, the total number of jacket cells is 16. The opercular cells or lid cells are absent. Anterior to the jacket of neck, a single dikaryotic neck canal cell is present. At maturity, the ventral canal cell and neck canal cell degenerate and form a sugary mucilaginous substance rich in malic acid. This substance exerts pressure at the distal end of archegonium, due to which the distal tier of neck cells is ruptured and the neck cells are far separated to establish a passage, and the substance comes out in the form of a droplet. The fertilization takes place in the presence of water between the lower surface of prothallus and soil. Malic acid of archegonial secretion helps in attracting the male gametes by chemotaxy. The male gametes enter the archegonium and one of the male gametes fuses with the egg to form the diploid zygote with its own wall. Fertilization is oogamous. Out of several archegonia of the prothallus, usually only one is fertilized. In some cases, even 2 or more archegonia may be fertilized. With the act of fertilization, the growth of prothallus is arrested. The archegonial venter forms a cap-like cleptra around the fertilized egg. The zygote develops into an 8-celled embryo that later develops into the adult sporophyte. The first division in the zygote is vertical and forms 2 cells. The second division is also vertical but perpendicular to the first division to form 4 cells. The third division is transverse to form 8 cells. At this 8-celled stage, the embryo gives out the primary root from its lower tier region that pierces the cleptra and establishes contact with the soil. Later, the first leaf emerges through the apical notch of the prothallus. It turns green and performs photosynthesis. The shoot grows slowly to become an underground rhizome. At this stage, the primary root pierces, and in its place, many adventitious roots arise from the rhizome. Now, the life cycle of genus Pteris. The dominant stage in the life cycle of Pteris is the diploid independent sporophyte that produces spores in its sinosauria. The spores germinate and develop into the haploid gametophytic phase, the prothallus. The prothallus is highly reduced, short-lived, and independent. It reproduces sexually with the help of Antheridia and Archegonia, wherein are formed spermatozoids and egg cells respectively. Fertilization of egg cell by a spermatozoid forms a diploid zygote which develops into the normal sporophyte. Hence, the life cycle is diplohaplontic, since both gametophyte and sporophyte are morphologically different. Its alternation of generation is described as heteromorphic. We have discussed over here morphology, anatomy, and reproduction of genus Pteris. Dear students, we have also discussed life cycle of Pteris. Thank you very much. God bless you.